The physical deterioration of the liver follows a predictable path of escalation. But under specific chemical conditions, the expansion of fibrous scar tissue stops dead. To understand why, researchers analyzed data from the UK Biobank, tracking 355,000 patients over a 13-year period to record instances of chronic liver disease progression. This chart highlights the statistical shift discovered. One cohort demonstrated a 32% lower risk of cirrhosis, a 47% lower risk of liver cancer, and a 42% lower risk of liver-related death. Patients achieving these results simply consumed coffee daily. This data indicates that coffee functions as a targeted pharmacological agent, requiring an explanation of the biological processes involved. The hepatoprotective effects observed in the study are not binary. They follow a dose-dependent relationship. This graph shows the clinical response. Protection scales with consumption, reaching its maximum efficacy at 3 to 4 cups per day. Clinical blood tests support this, showing a direct inverse correlation between consumption volume and elevated liver enzymes, specifically ALT and AST. To find the drivers of this response, we have to look past the clinical data and examine the liver's underlying cellular pathology. Liver deterioration is driven by 3 biological mechanics: steatosis, which is fat accumulation, fibrogenesis, the physical scarring of tissue, and oxidative stress, leading to carcinogenesis. Tracing the 13-year results requires identifying how specific bioactive compounds in coffee physically counteract each of these 3 drivers. We begin at the microscopic level to see how coffee affects fibrogenesis. Within the liver architecture is the hepatic stellate cell. In a healthy organ, this cell remains dormant, regulating blood flow and storing vitamin A. When the liver sustains damage, these stellate cells activate. They expand into myofibroblasts and begin secreting dense collagen fibers to wall off the injury, eventually forming permanent scar tissue. This is where the first bioactive compound, caffeine, enters the cellular environment. Caffeine physically docks into adenosine A2A receptors on the stellate cell membrane, blocking incoming activation signals. Inside the cell, this inhibits focal adhesion kinase, halting actin synthesis. This triggers the downregulation of signaling proteins, CTGF and TGF-beta. By interrupting that signaling pathway, the biological instruction to produce collagen is silenced. This receptor blockade explains the 32% reduction in cirrhosis identified in the UK Biobank data. Beyond structural scarring, the liver also fails through metabolic fat accumulation, or MASLD. Moving to the hepatocyte, the functional cell of the liver, we can see lipid droplets accumulating under metabolic stress. The defense against this accumulation relies on a second set of compounds, hydrophilic polyphenols, called chlorogenic acids, or CGAs. These CGAs easily dissolve in water, allowing them to rapidly cross the hepatocyte membrane and enter the intracellular fluid. Once inside, the chlorogenic acids initiate a sequence that activates the AMPK enzyme. Activating AMPK causes a downregulation of the liver's lipogenic enzymes, shutting off its fat-producing machinery. Simultaneously, this pathway improves the cell's insulin sensitivity. The combination of these 2 shifts clears the lipid droplets by halting de novo hepatic lipogenesis. This cellular shift is why coffee consumers demonstrate the stabilization and prevention of metabolic fatty liver disease. That leaves the observed 47% drop in hepatocellular carcinoma. Inside the nucleus of the hepatocyte, oxidative stress causes DNA fragmentation, the primary driver of carcinogenesis. The interceptors here are a third class of compounds, hydrophobic diterpenes, known as cafestol and caweol. These molecules stimulate the antioxidant responsive element pathway via NRF2 induction, sending a signaling cascade into the nucleus. This induction upregulates the production of phase 2 detoxifying enzymes, specifically glutathione S-transferase. Simultaneously, it suppresses phase 1 enzymes that activate carcinogens. This creates a functional cellular defense grid. Unstable reactive oxygen species are neutralized before they can damage the cell's DNA. This dual-phase enzymatic shift provides the systemic protection against liver tumors seen in the epidemiological data. Large-scale data shows that decaffeinated coffee also demonstrates significant hepatoprotective properties. We have identified 3 distinct pathways: fibrogenesis blocked by caffeine, steatosis reversed by CGAs, and carcinogenesis suppressed by diterpenes. The chemistry of decaffeination removes the caffeine molecule, but the polyphenols and diterpenes survive the process. Decaf remains effective at preventing fat accumulation and oxidative stress, though it lacks the specific A2A fibrogenesis blockade. This confirms that coffee's efficacy is the result of a multimodality of compounds operating on parallel cellular tracks. Pulling back from the microscopic models, we can see the systemic effect on the human liver. The dose-response curve from earlier reflects the saturation points of the A2A, AMPK, and NREF2 pathways. 3 to 4 cups a day is the point where these specific cellular receptors reach full biological saturation. These molecular pathways are effective adjuncts, but they do not replace established treatments like antiviral therapies, weight reduction, or lipid management. The clinical reductions in cirrhosis and cancer observed by the biobank are the result of 3 distinct targeted molecular blockades operating daily.